Hey everyone and welcome. I'm really excited to be here. This is the session on increasing your cell culture harvest yields utilizing centrifugation. So we're talking today about bioprocess. My name is Angela Carnwright. I'm the Senior Manager of E-Business Marketing, and I'll be your moderator for many of the live sessions this week. For the presentation, we're going to share the slides and then go live on video for Q&A. Just a reminder for this event type that the Q&A is actually through the chat function of Q&A. You can submit your questions anonymously or with your name attached, and I will be the moderator in the background, and I'll help get those questions out to the speakers. So today we're going to talk a little bit about how centrifuges work in the bioprocess workflow, and then how you can use them to optimize your yields in that process. So today our speakers will be TT Hong. He's the commercial manager for the floor model centrifugation products. He's been with Thermo Fisher Scientific for over 14 years in various roles, selling and supporting centrifugation products. With TT is Benoit Limo. He is the process application specialist for the floor model centrifugation products based out of France. He's been with the Thermo Fisher Scientific for over 20 years in various roles, interacting with bioprocess users and centrifugation products. I'm really excited for this talk today. I'm so glad that you could join us. Please sit back and enjoy. And if you have any questions, again, please go into the Q&A and post them in there. Thank you so much. And I'll hand it over to TT if you want to share your screen, please.
All right. Thank you, Angela. Can you see the screen?
Yep, it's up.
Great. Welcome, everyone, and thanks for attending our session today. We'll go ahead and jump right into it. We'll begin today by identifying applications where centrifugation can play an important role in the bioprocessing workflow. We'll follow that up by reviewing the important considerations when deciding what method of separation to utilize during the harvest stage. Finally, we'll finish up today's talk with Thermo Fisher Scientific centrifuge product offerings, which will help you attain these higher harvest yields. There are three stages of the bioprocessing workflow where centrifugation can play an important role: the harvest stage, clarification stage, and the final stage, where we'll call it the purification, concentration, or polishing stage, depending on what you're looking to do with the product of interest. But for today's presentation, we'll focus only on the harvest stage, since we're looking to find out how to increase harvest yields. Before we proceed any further, let's take a minute to ensure everyone understands what bioprocessing is, the workflow, and the various applications. Benoit, will you please do us the honors in going over each of these?
Yeah, sure, TT, and hello, everyone. So, what is bioprocessing again? This is a reminder for everyone that bioprocess is a specific process that uses a complete living cells to obtain the desired products. The cell manufacturing processes can be separated into an upstream and downstream process. On the schematic, you can see the workflow, a typical bioprocess workflow, with the upstream, with the cell isolation, the cell cultivation, and the cell expansion. Once the expansion is complete, then the downstream process will separate the cell or the supernatant, which is called the harvest, and then the material is clarified, purified, and concentrated, and then the final product of interest is then formulated. The upstream in the bioprocess is the entire process from the early cell isolation and cultivation to the cell banking and culture expansion of the cells until collection of the live cell batch. And the bioprocess downstream is when the live cell batch from the upstream stage is processed to meet the purity and the quality requirements. It is divided into successive steps. the separation of the biomass from the supernatant. This is called the harvest. When the intracellular product is kept into the cells, then the biomass, the cell palette, is the harvest. When the product of interest is extracellular, then the supernatant is the harvest. Sometimes the cells are disturbed and the product of interest is released from the cell. The supernatant containing the product of interest is then clarified to remove the cell debris, and then purified and concentrated, polished, and then final formulated and packed. Let us remember what are the typical applications in which centrifugation are used in the downstream process. For example, the cell therapy manufacturing is using the centrifuge to harvest the cells and then to concentrate. The whole cells are separated from the culture media, and this is requiring a little G-force between 500 to 1000 G. In the case of the gene therapy manufacturing, the centrifuge are used in a different step of the process. It can be used for the preparation of the DNA vector, requiring the separation of the microbial cell from the culture media. This is usually a G-force between 5,000 to 10,000. And then once the cells are transfected, usually the centrifuge are used to separate the cell from the supernatant. And most of the case, the supernatant is the harvest containing the product of interest. And then the centrifuge can also be used to clarify the supernatant, requiring a little bit more G-force, and then it can also be used for the final polish, requiring ultra-speed centrifuge. In the example of monoclonal antibodies manufacturing, the centrifuge is again used to separate the supernatant from the cell and harvest the supernatant with a speed of about 500 to 5000 G. And then the supernatant is clarified with centrifuge to remove the cell debris with a G-force about 10,000 to 20,000 G. In the case of mRNA viral vaccine manufacturing, again, the centrifuge can be used to prepare the DNA vector and separate the microbial cell from the culture media, and then separate the supernatant from the cell palette, and then again clarify the supernatant at various G-force depending on the size of particle and the step of the workflow.
Thank you, Benoit. Now we'll review the important considerations in order to increase your harvest yields. Benoit, will you begin by explaining how shear damage is important and why it should be considered?
Yes, of course, Titi. Let's investigate a little bit about the shear damage control and how this affects the yield in the cell culture harvest. So as a fact, some cells are fragile and delicate. The separation can cause stress on cells, leading to shear damage. When a cell is damaged, the cell viability decreases, the cell debris and intracellular product contaminate the supernatant, leading to reduced overall yields. So to increase the cell culture yields, it's then important to minimize the shear damage effect. Let's evaluate how 4 different separation techniques can control the shear damage. Let's start with the tangential flow filtration. We feel that the control of the shear damage is difficult, and we evaluate the risk to be high that the damage of the cell will affect the yield of the cell culture harvest. And this is due to the multiple passage flow of the cell over the filter, causing shear and damaging the cell, and cell debris contaminating the supernatant and losing shear and losing cell, therefore affecting the overall yield when using the tangential flow filtration technique. In the deep flow filtration technique, we evaluate the control of the shear damage to be medium, and we are thinking that the risk is moderate, that the cell shear damage will occur. And this is primarily due to the pressure differential across the filter pressing on the membrane of the cell. In the case of the continuous flow centrifugation, we are thinking the control of the shear damage is medium, and we evaluate the risk to be moderate, that the cell shear damage will occur due to the union seal design. Some shear effect might occur in the area where the rotating part is coming in contact with the static portion in the continuous flow centrifugation design. In the case of batch centrifugation, we are thinking the control is easy and the risk is low. And the risk is mainly due to the pressure differential caused by the G-force inside the container, which is easily controlled by adjusting the acceleration, the deceleration rate, as well as the rotation G-force.
Thank you, Benoit. So we'll use this table here to compare the various separation methods and their effectiveness against the various controls we'll be describing today, and I'll be adding to this as we move along in the presentation. So summarizing what Benoit just described, batch centrifugation seems to be the leading choice, whereas tangential flow filtration is the least effective method when it comes to shear damage control. Next is clogging. Clogging is another concern that ultimately affects harvest yields. So Benoit, will you please help explain why it's important, what methods are prone to clogging?
Yes, Titi, let's look into the clogging control. Some separation methods can clog during the process, and a clogged process results in a halt of production. When the process is stopped, the batch might be rejected and the overall yields is reduced. To increase the cell culture yields, it's important to avoid clogging during the process. Let's evaluate how the 4 different techniques shown here are controlling the clogging in the cell culture harvest. In the case of tangential flow filtration, we feel the control is medium and the risk of clogging is there is a risk of clogging when the cell density is high. In the case of deep flow filtration, we are thinking the control is difficult and that there is a high risk of clogging when the cell density is high. Here we can understand that the cells are pressed against the filter, and there is a point that the filter will not be able to flow through the liquid, and that the process will have to be stopped. In the case of continuous flow centrifugation, we are thinking the control is easy and the risk is low of clogging even though the cell density is high. In the case of batch centrifugation, we are thinking the control is easy and there is no risk of clogging.
Now, referencing back to our table again, the risk of clogging is highest in deep flow filtration, followed by tangential flow filtration. It's not much of a concern, as you can see in what Benoit had just described when utilizing continuous flow or batch centrifugation. Next, the next consideration is void volume control. And Benoit, will you go ahead and go over void volume control and why it's important to consider this?
Yes, of course. The control of the void volume is also key in order to optimize and increase the yield in the cell culture harvest. Some separation methods can hold up some volume, and that hold up volume is unprocessed. And when some volume is unprocessed, the overall yields is reduced. So in order to increase the cell culture yields, it's then important to minimize the void volume. Let's evaluate how the 4 different techniques are able to control the void volume. In case of tangential flow filtration, we are thinking the control is medium, and there is a medium risk of a void volume. In a deep flow filtration, the same applies as well. In a continuous flow centrifugation, we are also thinking the control is medium, and also there is a medium risk of void volume. This is mainly related to the nominal volume of the continuous flow rotor. In the case of batch centrifugation, we are thinking the control is easy and there is no risk of void volume. The total supernatant can be recovered as well as the total amount of the biomass cell palette.
Recapping again what Benoit just described, the avoidance of void volume is the best when considering batch centrifugation, and all other methods are simply not as effective. Moving along, our last consideration today is separation efficiency. And Benoit, will you continue on with this control?
Of course, Titi. The control of the separation efficiency is the fourth control we will examine today. And this is also key to increase the yield in the cell culture harvest. The separation method, some separation methods do not produce a dense cell palette. The lack of a dense cell palette could mean inefficient separation of the supernatant and pellet, resulting in a decreased harvest yield. To increase the cell culture yields, it's important to optimize the separation efficiency to generate dense cell palette and clear supernatant. Let's evaluate the 4 different techniques in the control of the separation efficiency. In case of tangential flow filtration, we estimate the control to be difficult, and this method is resulting in a slurry cell palette and a clear supernatant. In order for this technique to work, the cell palette has to be kept as a slurry, moving and flowing through the filter. When the cell concentration becomes too much, then this process is ending. So this indicates that the cell palette is still containing some supernatant and the recovery is not fully optimized. In the case of deep flow filtration, We evaluate the control to be easy. This method is delivering a clear supernatant, but the cell palette is kept trapped inside the filter and cannot be harvested. So this method is enabling the harvest of a supernatant only and waste of the cell palette biomass. In the case of the continuous flow centrifugation, we are thinking the control is easy and it's possible to obtain a dense cell palette and a clear supernatant. In the case of batch centrifugation, the control is easy, and this method is delivering a clear supernatant and a dense cell palette biomass.
Thank you, Benoit. In reviewing all of the considerations that were just described, I think it's safe to say that we can infer that batch centrifugation is the best technique to use overall to increase your harvest yields. So now in our final topic, let's talk about what solutions are available from Thermo Fisher Scientific for someone wanting to consider batch centrifugation in their bioprocessing workflow. I'll take the next few minutes to go over the centrifugation equipment and consumable offerings we have. If you recall earlier, we identified 3 stages of the typical bioprocessing workflow where centrifugation plays an integral part. For the harvest stage, we have our Bio16 bioprocessing centrifuge with industry-leading capacity and ergonomics. In the clarifying stage, we have the BIOS-A bioprocessing centrifuge that provides increased performance features in comparisons to the Bio16. And then we have our innovative and popular Lynx SuperSpeed centrifuge platform. When looking to purify, concentrate, or polish, we have the WX Plus Series Ultra Centrifuge or the CC40 Industrial Centrifuge platforms. But for those looking to do batch centrifugation, we recommend either the Bio16 or the BIOS-A centrifuges as ideal choices. Now here's a table that briefly goes over the general specifications of these 2 centrifuges. Listed here, you'll find the types of containers available, the max capacities and max speeds for these centrifuges, and what cell types can be processed using these various centrifuges. But focusing in on the Bio16 centrifuge itself, I want to briefly mention 2 new consumables we've launched this year. The first is our SteriBios 2000 milliliter sterilized centrifuge bottles. And the second is our CentriPack system, which is a unique, one-of-a-kind bioprocessing container designed to be centrifuge ready and takes you from reactor to harvest to purification, all in a single-use sterilized closed system. For more information on these containers and equipment, I recommend that you reach out to your local Thermo Fisher lab equipment representative, or you can simply go to thermofisher.com where you can get more details on bioproduction solutions, centrifuges, application notes, and more. Thank you all for your time today.
Hey everyone. So there are a couple of questions here. So I'm actually going to send you guys live. I think I can only send one at a time. So TT or Benoit, do you want to go on your video feed and I'll send you live?
Okay, very good.
Great, thanks. Okay, so the first question is, what single-use centrifuge-ready consumables do you carry for harvesting? So which consumables match up with some of the products we talked about? Can you remind us?
Yeah. Benoit, you want to answer that, or do you want me to go ahead?
Yeah, go ahead, Titi.
Yeah. So as I had mentioned, the 2 new consumable offerings that we launched this year are SteriBios 2-liter sterilized bottles. We launched this bottle to finish out the family of our 2-liter bottles for the Bio16 centrifuges, where you can maximize, bioprocessing capacities utilizing this consumable. And then we have our unique product, the Centripack system, which is that single-use, sterilized, centrifuge-ready, and closed system consumable. This product was developed in conjunction with our partners over in the bioprocessing group, and it's a great solution for harvesting in the bioprocessing workflow.
Awesome. And one of the other questions as a follow-up are there customers currently using the Centripack? If we needed references or ideas on how people are utilizing it in their applications.
Yes, we do. We just recently launched this product in the US. I think we're in the midst of launching in Europe as well. So, but here in the US, we have a customer in Minnesota that has been utilizing the product, and they, from the feedback I've gotten, it's been a great success for them.
Thank you. So I have another question here too. What are other considerations that one must keep in mind when deciding on what technique to utilize?
Yeah, I can answer this one, Titi. So the other consideration will be the type of cells, the density of the cells, the turbidity of the cell culture, the volume to process, the time required to process this entire volume, with whether the supernatant or the biomass is the harvest, the cost of initial investment, the cost of the operation, and the serviceability of the system in place in the step of the process. So many other important consideration to be considered.
There's one more that came through, and the question was more on the R&D side. So the question is more, is there a place for these bioprocess centrifuges in an R&D workflow, or is it just more in a kind of when you have a product that you're producing for a company?
Yes, so these techniques works at whatever scale of the process you might be. So it can be a R&D process, a pilot process, or commercial process. And Thermo Fisher, we offer different kind of solution for harvest system that fit the different scale of the process, and R&D is one of it.
Perfect, thank you. And I have one final question that's come through. The question's around environmental sustainability. There is some focus on that in one of our sessions later this week. Is there any aspects of this bioprocess centrifuge workflow that would be considered green or sustainable? Maybe the size of the batch, just, I don't know, it just kind of.
I think the Bio16 batch centrifuge is designed with the green aspect in mind. The rotor has a windshield, reducing the total power consumption and the heat emission. So the R&D team worked around the saving the energy, reducing the power consumption of the overall system. And also the container itself. In case of the bioprocess, it's important to have sterilized, clean, and traceable container. And working with a single-use solution might also reduce the consumption of the cleaning and sterilization effort, and bringing a greener solution to the market.
Excellent, thank you. Sounds wonderful. So I will remind everybody, there's no more questions at this point, but I will remind everybody that there is the Virtual Solution Center. So if you go to the left-hand navigation in the event platform itself, there is the Solution Center with our live virtual concierge. If you do have questions that you would like to address one-on-one with TT and or Ben Benoit, then you can go over to the virtual concierge. It will link you through to a normal Teams meeting that will give you the ability to share audio and video if you like, and you can check in with the team there. So there's always a person during normal event hours. There's going to be somebody there manning that booth. So while you're there, fill out your passport to science, check out the Bio 16, which is on display in our future lab. And I thank you all for joining us today. It's been fun.
Thank you. Bye-bye.
Thank you.